Hello everyone, welcome to our channel. So as we have already finished the translation process in our last video, so today we will discuss about the important antibiotics and toxins which are involved in protein synthesis inhibition. Basically this video will be very helpful for the CSI and Life Science NEET aspirants and the NEET JE aspirants, and all the life science students will get benefited through this video. So let's get started. First, the antibiotic is streptomycin, which targets cell is prokaryotes. It binds with the 30S subunit and causes the misreading of mRNA at very low rate. Thus, it causes the protein translation inhibition. Next is tetracycline. The target cell is prokaryotes. It binds with the 30S subunit and interfere with the amino acid tRNA binding. So if the tRNA cannot bind with the ribosome, so automatically the protein translation will get inhibited. Next is chloramphenicol. The target cell is prokaryotes. It binds with the 23S rRNA of 50S ribosomal subunit. So as we all know that the 23S rRNA is responsible for the peptidyl transferase activity. So if the antibiotic is getting attached with this rRNA, then automatically that peptidyl transferase activity will get inhibited and no peptide bond will form. Thus the polypeptide chain can't be formed, so the protein synthesis will be inhibited. Next is erythromycin. The target cell is prokaryote. It binds with the larger subunit, that is the 50S subunit, and it inhibits the peptide chain elongation. The next is fusidic acid. The target cell is prokaryotes. Basically, it inhibits the EFG factor. EFG binds with ATP, get activated, and thus it inhibits usually it causes the ribosomal translocation. But when fusidic acid come and it inhibits the EFG, then no ribosomal translocation takes place and the protein synthesis got inhibited. Next is puromycin. This is an interesting antibiotic which is the structurally it is similar with the 3' end of amino acid tRNA, as it is structurally same with the amino acid tRNA end, so the ribosome easily accept the puromycin, and then but the puromycin bind with the ribosome through amide bonding, whereas usually the tRNA used to bind with ribosome via ester bonding, which is easily cleavable, but the amide bond is uncleavable. So in that way the puromycin get fully attached with the ribosome and no further peptidyl transferase take place and the ribosome falls off, and this is work in eukaryote and prokaryotes both cell. Next is diphtheria toxin. Diphtheria toxin come from the diphtheria bacterium, and it is an exotoxin, and it inhibits the protein synthesis in eukaryote by inhibiting the elongation factor, that is EEF2, by ADP ribosylation. Means it EF2, this elongation factor permanently bind with the ADP, thus it becomes permanently inactivated, and this is means the a part of the diphtheria toxin is causing the mainly the inhibition of protein translation, and the target cell is eukaryotes. Next is ricin. This is also an toxin. It comes from the caserbin, that is Ricinus communis. It inactivates the 60S subunit by depurination of the 28S rRNA adenosine. Next is cyclohexamide. The target cell is eukaryotes. Peptidyl transferase center of the 60S subunit is targeted and inhibited. Next is paromomycin. The target cell is prokaryotes, and it actually binds nearby place of the A site of 30S ribosome, and it causes the interfere in codon and anticodon interaction. If the codon anticodon cannot interact properly, then many error can happen during translation, and by decreasing the selectivity of codon anticodon pairing, it causes the inhibition of protein translation. So this is all about the antibiotic and the toxins which causes mainly the inhibition of the protein translation in prokaryotes and eukaryotes. Hope this video is very helpful for you all. Thank you so much for watching this video.